Bueno, buenos días a todos. Hoy es la segunda ocasión en que nos reunimos de esta manera por vía remota. Hoy tenemos la presentación del doctor Ferhat de la Joya Institute for Immunology. El doctor Ferhat obtuvo su licenciatura en Ingeniería Computacional y Matemáticas por la Universidad de Ankara, en Turquía. Y después se decidió un poco por la biología y entonces realizó un doctorado en la Universidad de Florida en análisis de redes genómicas que regulan la actividad metabólica y la expresión génica en un análisis comparativo en múltiples organismos. Este trabajo le valió realmente la obtención de varios reconocimientos y le permitió después realizar un postdoctorado en el laboratorio del doctor William Noble en la Universidad de Washington en Seattle, en donde se empezó a orientar hacia lo que está haciendo ahora, que es estudiar la estructura 3D del genoma. Y la definición de su trabajo en realidad es justo eso, es ver más allá de las secuencias lineales del DNA para entender cómo la arquitectura tridimensional del genoma participa en una gran variedad de procesos biológicos que van desde la adquisición del fenotipo maligno de las células cancerosas hasta la regulación del ciclo de la vida de Plasmodium, el agente causal de la malaria y muchas otras enfermedades, en particular ahora que está en la Joya Institute for Immunology, en cosas asociadas a la inmunología. Como verán ustedes, su trabajo es muy completo y incluye justamente un gran análisis computacional de las secuencias y basado en resultados experimentales. Y realmente es muy, muy interesante lo que hace y muy didáctico también. Ferhat, thank you so much for joining us and for giving the class yesterday for the students and today for joining for the seminar. And thank you so much.
Thanks a lot for the invitation. It was a pleasure yesterday. Today it's going to be much less interactive, I think, but I hope people on the other side of the line, they will enjoy what they're hearing.
Yes, before you go, look, what I would like to remind people is that you can ask your questions on the YouTube channel. And then we will read them on the Zoom because it's a composite system for this.
Yeah, so you will keep an eye on the channel and everything, I hope. I'm just going to focus on the. Okay, all right. Thanks a lot again. This is a great opportunity for me to strengthen ties with our neighbors to the south. As Yvonne mentioned, I'm at the La Jolla Institute for Immunology, but I come from a computational background. So we are kind of like harmonizing the two, that the environment that we are in, which is a great immunology institute, and then the experience that we have and the training we have in developing computational methods in the context of immune system. So today I'm going to talk to you about maybe 1 or 1.5 projects. So mainly I want to focus on the gene regulation through distal interactions in the human genome in the primary human immune cells that we want to focus on. So as you may see, I think my lab is, we are interested in a lot of different contexts and diseases of how we can apply what we do the best. And the only reason why we are able to do or why we are able to contribute, I hope meaningfully, to a lot of different disease scenarios is that is the development of computational methods. So it's an exciting area to be in, and it's also very stimulating in terms of the things that you can accomplish. I'm not going to talk about all of these, but I just wanted to show you that the computational methods and our efforts towards that is what helps us apply it, or what helps us contribute to these different topics. So first I'm going to start with the chromatin structure. This is our core interest. So at just the basic science level, we are interested and intrigued by the fact that the genome and the chromosomes, our DNA, really we think of it as a linear DNA sequence, but more and more I think we shifted our understanding and thinking to the 3D structure. how this almost 2 meters of DNA per cell, which is very long, and for human cells, this fits into a tiny, a few micron wide nucleus, right? So there is an immense amount of packing, and that packing is not random, and that is meaning, and disruptions or the differences in this packing may mean health or disease, right? So again, like you can think of this linear genome, you can look at it this way, and then the enhancers, you should think of them as the switches, and then the genes, you can think of them as lights. So you look at the sequence, and then you wouldn't guess that that switch might be controlling that light. But if you looked at the 3D genome, it will be obvious that their physical proximity makes them more likely to be like two things that are functionally connecting with each other. So these are the kind of things that we like to study. So the field essentially, like there were imaging and other techniques in the field to really understand how the chromatin is organized in the nucleus, but the whole high throughput efforts in the field started maybe by now, like more than a decade ago, when the Jaap Dekker's group came up with the experiment that's called HiChip. So what this does is, you have your cells, they have their nucleus, and then within the nucleus, the DNA is folded in whatever structure it is folded in, right? So you kind of want to get a sense of it. So how you do that is that you take those cells and nuclei, and then you do something called formaldehyde cross-linking, which essentially looks like this, conceptually at least, that holds together the different molecules such as DNA interactions, DNA-protein interactions, and all of them, that holds them in place for a while, such that you kind of like took a snapshot of the 3D structure, and then you start doing some molecular fiddling with the system, trying to get a readout of it. So what you first do is you introduce restriction enzymes, and then they will digest the genome from specific sequences, or 4 base pair or 6 base pair cutting regions in this case. And then that will lead to these kind of fragments, that 2 DNA pieces that may or may not be linearly close to each other, but that are certainly 3-dimensionally close. They are going to be held together by the cross-linking. So then you can do a few other things. You can repair these ends, and then you can put like a biotin mark here and so on and so forth. But at the end of the day, you can get these circular products. But circular things are not great for sequencing, so you kind of cut them again with something else, another restriction enzyme or just sonication. So that gives you these fragments that are chimeric. And by that I mean that this is a linear product, but one side can be from one part of the genome, and then the other side can be from another part that might be quite far away in the linear distance. So then it's easier. Then once you have that, then you can do paired-end sequencing, and then read a little bit from this side, read a little bit from that side, and then the bioinformatics parts come. Then you align these reads to the genome, find where they are coming from, and then start creating these things that we call chromatin interaction maps or contact maps that tell you if the red is darker, these 2 regions tend to be closer together. If you have a light color like this, they tend to not come close to each other in the 3D distance. So I hope this is clear, because that's kind of like the whole starting idea of everything else, right? So this is a way of taking a nucleus, trying to get a proxy for the distance between each pair of regions on the DNA, and then from that all our business starts. So if you looked at the human genome and then just an average HiC data, or just one HiC data from a cell line, the first thing that you are going to see is that the chromosomes obviously tend to fold within their own corner and mingle less with the other chromosomes. So that you are seeing is called chromosome territories, right? So that has been known and seen in the literature for quite some years already. HiC confirms it. This is at the 1 megabase resolution, meaning that there are windows of 1 megabase DNA region here and then 1 megabase DNA region here, and how many times they are interacting is the heat. If you zoom down a little bit more, if you go to a finer resolution, then you will start seeing these what we call plaid pattern that corresponds to these chromatin compartments. So what that means is that the regions that tend to interact with each other and that tend to be on the nuclear lamina, the kind of like outer layer of the nucleus, they tend to be inactive, and then they tend to correspond to these enriched dark reds here, meaning this region is a B compartment, this region is a B compartment, they tend to interact. And then the other side is A compartment, which is active chromatin, expressed genes and active chromatin environment that tends to be located towards the interior of the nucleus. And then you zoom in a little bit more. We have some work about differential analysis of it. I don't think I will have time to talk about it. So then you take a region and then zoom in a little bit more at a higher resolution. Then now you start seeing these square shapes that are appearing on the diagonal. So these are what we call domains. So these are what we call topological domains. These are regions where interactions tend to occur more frequently within, but not across the two different domains. Right? What it looks like is it's like this. It's like a folded chromatin that is demarcated by some marks, which we will talk in detail about. And within these squares, sometimes you also see some enriched dots. So these would correspond to, even within that domain, there are still preferential proximities between different regulatory elements. These can be enhancers and genes and gene promoters and so on and so forth. So the other dots, they correspond to these regulatory interactions that generally happen within the domain. So my lab has been highly interested in these loops, either the ones that are demarcating these kind of domains, or even more the ones that are happening within a domain between specific regulatory elements. So we have published a whole bunch of papers on that, and most recently we I think I'll talk about this. We published the image processing based method to identify these bright dots. And then the other project that I'm going to talk about is actually just accepted at Nature Genetics, so it's going to appear hopefully within 2020. It's about, again, like finding these loops in a high throughput manner in human immune cells that we are very interested in. So this is a paper that I mentioned, the paper where we use image processing. So generally how people have approached, including us and my earlier work, to finding patterns out of these contact maps is that you have it at a specific resolution, and then you try to, pixel by pixel, you ask whether this pixel is an interesting one compared to background and so on and so forth, right? But that has two problems. That first of all, these interactions can happen in multiple different scales, which is shown here. So it might be a two regions that are tightly interacting, but not, like the broader regions around them. Or it might be something like that involves broader regions, but the interaction isn't as strong, right? So the previous methods were missing this information. So this method is trying to address that. And then the other the other thing that they were missing is that these interactions, they happen at you can be looking at different bin sizes and finding some of them and not finding some others. So this was the whole idea. And then we have shown that compared to highly used methods in the field, such as HiChIPs, in deeply sequenced data sets, we are finding almost twice the number of loops. And these loops, by and large, contain what HiChIPs is finding. So we are finding what was found in the literature plus maybe another 10,000, which we then show they are important and interesting ones. And then if you look at the reproducibility between two replicates, we are getting up to 80%, which is very high actually for the literature, for the field of high-C interactions, showing that our method is quite robust. So this is now, hopefully people will use it for high-C and improve the resolution version of it that's called micro-C. And we actually have been answering user questions since the publication, which has been great for us. So, but in general, I think that what's like interesting scientifically is that how are these loops formed, right? So I kind of want to give a little bit of background on that so that people have a better understanding before I go into our own work. So again, like back in 2014, when the high-C resolution reached the point where we can talk about looping, what the people have shown, the Erez Lieberman's lab has shown, is that the at these loop boundaries that there exists a insulator protein called CTCF. It's highly enriched, and it's binding in a specific configuration, in a convergent configuration, essentially. Like it binds one way on one side and the other way on the other side. So that was just an observation. Right? But of course, observations like that trigger, you know, hypotheses and models. So the model that came out of this, again by the Rao lab and Leonid Mirny and a few other people around the same time started coming up with this idea of loop extrusion. So what that means is that this is your DNA in an unfolded manner you can think of. Right? So the idea is that there are these ring structures that might be getting loaded onto the DNA. And in an active ATP-dependent manner, they may be extruding the extruding the chromatin, extruding your DNA. And then they keep doing that, and growing this domain up until they reach something like a stop sign. And that something is CTCF. On one side, it reaches that. On the other side, once it reaches a convergent one, this is going to be stopped. This is going to be like established as a chromatin domain. It's going to look just like this, like a square with a dot on top of it. Right? So the nice thing was that they were able to come up with a computational model that just looks at the CTCF binding across a given cell type and predicts which one's prediction? Okay, this one. That predicts through the computational model how the chromatin's folding should look like. And then they were able to show that by and large looks like what you would measure by the experiment. And then the more interesting part was they could go look at the CTCF binding in these specific regions. And through CRISPR, as you know, CRISPR just got the Nobel Prize, and it's been highly impactful in everyone's lives in our case. So through CRISPR, you can delete or you can even take and invert these CTCF binding sites, and then you can predict what would happen, and then you can do experiments and see actually that the model gets validated. So that was nice, at least that gave us some model to think about how chromatin is folding. So the follow-up work on that by many other people, first of all, it showed that this CTCF that we believe is at the boundaries of these domains, if you degrade it, then these loops and tads are mostly gone, right? But interestingly, there was not much of a change to the transcription itself, and there was really not much of a change to the compartmentalization, this general A and B chromatin compartments, even though the tads were lost. So around the same time, us and collaborators, we also looked at this, and then as you can see, we have shown that this doesn't have, CTCF depletion doesn't have any effect on something called replication timing, which is how fast your DNA replicates after each mitosis. So then when you look at the, that was the CTCF, and then I showed you these rings that are folding onto the DNA. So what are those rings? And then the, you know, the usual suspect was cohesin. And then people have also looked at cohesin and degraded it. And then the same idea, it eliminates these loop kind of domains, but it doesn't eliminate these kind of domains where it's mainly compartment. And again, little change to transcription. Then people start looking at how about the things that either release the cohesin from the DNA or that load the cohesin. So deletion of a cohesin release factor, in this case called WAPL, actually leads to stronger loops. The cohesin resides more on the DNA, and then loop structure is kind of like heightened in this case, as you can see. But it limits the compartmentalization. So that brought the idea that this domain formation and compartment formation is kind of like two, forces that are pushing each other, that are competing. So then a cohesin loading factor, so whatever the protein that loads cohesin into the DNA, if you kind of delete that, then this time you get a lot of compartments. These were all nice, but one paper that kind of has been really, really impactful in our understanding of all of these has come from all of these other papers came from the sequencing-based assays such as HiChip. But at the single cell level, which is where the things are really interesting, Xiaoyi Zhuang's group from Harvard, they came up with this really nice assay, imaging-based assay, where they can, for at least a given region, they are able to find chromatin interaction contacts in their intensities at single cell level. So what they have shown that by and large, if you have enough hundreds of single cells and kind of combine the data from them, then you get these domains just like in HiChip. But if you, for instance, use the same cohesin depletion strain that was used somewhere here, then what happens is these in the aggregate you get nothing, right? So that aligns with the cohesin loss experiment. So you have the TADs, you don't have the TADs in the depletion. But what was interesting is that the reason why you are not getting a signal in the bulk is it seems like it's mainly because not because there are no more squares on the single cells, I'm not showing here, but where they are appearing kind of is not synchronous anymore. So these boundary probability of this region being the demarcating region for the TADs, it was non-uniform. There were CTCF regions and cohesin regions in the non-cohesin depleted. And in the depleted one, it kind of becomes uniform. Therefore, you do not have a synchronized one, and then you kind of like lose this. So that really helped us think about single cell heterogeneity and enhancer promoter interactions and topological domains. So that's maybe a little long, but that was literature, how these loops are formed. So then comes our project. So the idea is that there are these genetic variants on the human genome that we want to know what their gene targets are, because they are in the non-coding genome, right? And then we have this really nice resource at the La Jolla Immunology, established by Vijay's lab, that we call DICE, so Database of Immune Cell Expression and eQTLs and epigenomes. So I mentioned this yesterday as well, right? So once the human DNA was sequenced, back like almost 2 decades ago, the whole idea was that, okay, this is great. Now we can take a cohort of people who are healthy and a cohort who has a specific disease, I don't know, type 1 diabetes or something that has genetic component, right? Then we will compare them, and then voila, we will find all the right things and all the right genes and everything, and then try to treat these people. But this hasn't been that easy, and there are many reasons for that. But one reason being a lot of the genome, if you look at this chart, a lot of the genome is actually not coding for genes. It's like 98% or so doesn't have genes in them, but we believe they are still functional. So when you look at these genetic associations coming from the GWAS studies, a big percentage of them come from non-gene coding regions. And then now you do not know where that specific variant maps to. Why is this important and is a big problem? Is that one example of why is coming from this really nice and old genetics, mouse genetics studies, actually, going from like a human phenotype such as polydactyly, where these kids would have like 6 or 7 digits rather than 5, It's a developmental disorder. And then the mouse has a similar phenotype. So what was known is that SHH gene, that the sonic hedgehog gene might be the driver of this, but what was not known is that what controls this gene's expression, right? So what they were able to find is that this 1 megabase, so that's 1 million base pairs actually. So this 1 megabase region in the genome, 1 megabase away region from the sonic hedgehog gene, seem to have mutations and polymorphisms that correlate with this disease and its occurrence. So in the mouse model also, when you go and delete the specific enhancer, as you can see, like the mouse doesn't have limbs, neither hind or forelimbs. So that tells us that if you, for a lot of cases, then you can look at the gene and then you can look around it, linear proximity of it, and then find what controls it, what's the control switch for that gene. But there will be cases like this that will throw you off, and the switch will be so far away in the linear genome. But then through later studies, actually through chromatin conformation capture, have shown that this enhancer is really actually looping and just right next to the SHH at the correct time, at the correct cell type. So then we thought we want to do this genome mind for all genes. And then we thought we want to do this for many different cell types so that we study cell type specific effects of these regulatory elements. Right? So the DICE database, the first version, was published back in 2018 with 13 plus 2, 15 immune cell types from 91 donors. And then you can go and nicely type your favorite gene, see how it's expressed across these different immune cell types, and then what are the eQTLs for each gene. eQTL meaning a SNP that differs between people, and then its difference correlates with the expression of a specific gene. So that's expression quantitative trait loci. So what we did, again, like the kinds of experiments that we like to do are quite expensive and quite laborious, actually. Like it takes one full week for Vivek to do these experiments. So we took a subset of the 91 donors, and then we took a subset of the cell types, 5 cell types and 6 donors. We have done this thing called HiChip, which I will mention about. But the whole idea is that for a gene, there might be a lot of eQTLs, but not all of them will overlap these regulatory regions, these enhancer elements. For instance, this one doesn't overlap with any enhancer region. And then the ones that overlap with enhancer regions, not all of them are going to be physically proximal to the gene promoter. So we kind of want to prioritize the ones that do interact with the promoter and do overlap with an enhancer region. So these are the 5 cell types, the most common, most prevalent cell types in the human, immune decomposition. And this is the assay that we have used from 2 different labs, PlaqueSeq and HiChip, back in 2016. They were published. So essentially, it's just like that HiC experiment, if you remember, but there is a step that's added. And if those of you who know ChIP-seq, there's an IP step that's added where you add an antibody for a specific thing that you are interested in. In this case, it's a histone modification. It's H3K27 acetylation for us. Right? And then you don't only get all the contacts genome, right, but you get the contacts where you are really interested, which is these K27 peaks. And as you can see, it looks different than HiCMap. A lot of white background, but then there are some regions where you have improved signal or increased signal. So this is nice. We get this at the end of this experiment. But then really what we want from this is something like these arcs where you look at the gene and then you connect it to the relevant enhancers that are potentially controlling the gene. Right? So here I'm just introducing, I guess, our method called FitHiChIP that does that takes this contact map and converts it into these kind of specific loops. And then this is an experimental data showing what are the enhancers that have an effect on this very important cancer-related MYC gene. And as you can see, we have a really nice correspondence. So that was our starting point. We kind of like, we had these. And then at the time, there were no really good methods that can take this and then turn this into these kind of loop calls. So this is, I guess, like for those of you who are computational biologists, this is a great example of where you can be a computational biologist working closely with the experimental labs and a specific topic of interest or biological problem of interest. And that, if you look carefully enough, that generally gives you opportunities to come up with new and different and nice methods, right? So this essentially, this stemmed from our necessity to be able to make sense out of these contact maps. And once you develop a method that way, then because you need it and you will use it, that is very highly likely to be needed and used by other people in the field, right? Then you immediately have an impactful method development effort. So that was the case in ours. I'm not going to go into the details of it, but essentially this whole pipeline can start from the very beginning and whatever comes out of the sequencer, and then it has a normalization part for two different kinds of background effects, and then it combines them like this distance dependence of the contacts, and then the peak height, the chip-seq peak height. It combines these two and then comes up with these arcs and these calls. Right? So a few things, of course, when we came up with this, we had to compare it to a few methods that were out there, and then a few more came and we were in the review process. So then we also had to develop ways to evaluate this in a genome-wide manner, in a non-specific or in an unbiased manner. So I'll show you a few of them. One is this previously experimentally validated data, right? The one that I showed you actually. So the one method that was out there, it was nice, it was stringent, but it was missing things. It was not sensitive enough. For this locus, for instance, it would only find this one specific loop from HiChip data. And then another method that was there when we came up with this, it was kind of very non-specific. It was calling a lot of things as loops. Some of them true, but some of them are unlikely, or they are indirect, right? So then we came up with our method that seemed to find the right things and so on and so forth, and assign them the strongest. So if I take the top 5, it would actually be like these 3 and a few more, right? So that was locus by locus analysis. The genome-wide analysis was, if I go into the literature for some cell types, there's a lot of other data. So if I go into the literature and then extract the other forms of chromosome conformation capture data, such as ChIP here with the cohesin antibody, or just general HiC with a very, very high sequencing depth that's been used before. If I took the intersection of the loops found from these two, which would give me a very stringent set, very confident set that these things are real, maybe around 5,000 of them. And if I looked at our method, and 4 different parameter settings of it versus other methods, one of them came in the meantime, how fast, like everyone, if I call 100 million loops, then of course I'm going to capture 5,000 things, but it's not going to be very specific. Right? So as each method is calling more and more loops, how many of these, or what fraction of these reference loops that we are capturing was the idea here. And as you can see, FitHiChIP for any given, if you call 10,000 loops, FitHiChIP and its all settings tend to recover more. If you call 20,000 loops, that's the same, right? So showing that we have both better sensitivity and specificity compared to these existing methods. And then one other thing is because I talked to you about the CTCF loops and everything, one other thing that we have shown is that through FitHiChIP, you might be getting thousands of additional convergent orientation, the right kind of orientation CTCF loops. Right? So this very stringent method is essentially getting about 4,000 loops from this specific data, with 80+% being the convergent CTCF orientation. Whereas our method in different settings, if you look at this one, for instance, it's getting 6,000 loops with 5,000 or so that are at similar fraction. So that tells you that there is still some more information to be squeezed out of this HiChip data that has been mainly missed by the existing methods, and that our computational method is making a difference there. Maybe one last bit that was interesting for us is that you can look at these loops at different resolutions, different bin sizes. You can look at them at 5 KB, you can look at them at 2.5 KB, any arbitrary bin width. But if you go higher resolution, your statistical power will, decrease. If you go, lower resolution, like bigger bin sizes, then you will have statistical power, but then you're not going to be really talking about specific enhancers and promoters. You will have big bins that will. So we kind of wanted to see whether our method was robust to this bin size selection. So as you can see here, if we did the loop calling at 5 KB bins versus 2.5 KB bins, by and large, most of the 5 KB calls, we will be able to zoom in and then find the specific half of this bin and then specific half of this bin that interact and explain this finding from the 5 KB. So we can resolve up to 80% of them to a finer resolution. And in some cases, it's a broader region on one side and so on and so forth. But by and large, this was what we are finding. So overall, I think then we were able to show and also convince the reviewers to publish that we have a balanced sensitivity and specificity in loop calling with FitHiChIP using HiChip data, better concordance with existing data, and then consistency across different resolutions, and better recovery of interactions that are identified by a whole bunch of other orthogonal methods and orthogonal experiments. So that was the computational part. I just wanted to kind of like almost go into this and then come back, because our problem, again, if you remember, we wanted to look at the immune cells from human population and then try to understand the genetic variations and how they are looping to their target genes, right? We generated the data, we needed a method, we developed the method, and then now that all these pieces are together, then we can really go into what we have discovered from this data. Right? So the very first thing that came out is that by using this physical proximity rather than linear proximity, we can completely redefine how people have been mapping eQTLs. So the idea goes like this, that the standard eQTL discovery, this is GTEx and a lot of big cohorts, when they take a population of individuals and when they take a gene, the immediate thing that's being done, and somehow it's set to 1 megabase threshold, is that you take a gene, mainly the promoter of the gene, and then you look at all the variants within 1 megabase to the upstream and then within 1 megabase to the downstream. And then you test every single variant here, however many hundreds or thousands of them, with respect to this gene's expression, to see if any of them associate with the gene's expression. Right? So we thought this is pretty arbitrary. I mean, everyone knows it's pretty arbitrary, but we thought, why do you care about the linear proximity so much, right? So you might have a gene, actually, that within 1 megabase, maybe this region is really not close, but very far apart in the linear distance, maybe you have a loop, right? So we said, what we are going to do is, instead, we are going to take this 1 megabase region, only test the things that are looping to the gene. Using that statistical power that we kind of set aside now, all the testing that we have saved, now we can go look at the ultra long range or extra, beyond 1 megabase distance for the associations. So that will allow us actually to find potentially new eGenes, genes that didn't have anything associating within 1 megabase, but do have things that are beyond, and also rewirings of the same or the previously discovered eGene to different QTLs. Okay. So this shows you that's kind of like what happened also. So that we can look at the gene, look at either direct looping partners of it or the indirect, kind of through two hubs. And then if you looked at them, then the there is about 20% out of all genetic variants that overlap these enhancer regions. Maybe 25% or so total either loop directly to the genome or to the gene, or indirectly to the gene, right? So about half of them you can immediately discard, saying they are not looping, so they are unlikely to be functional. The other 25% is things that are right next to the gene, so it's within this proximity, which makes all the sense. But then this allowed us. This is through all the variants that are on the enhancer regions. But if you look at all the eQTLs, all the SNPs, that's 200,000 or so per cell type. We are able to really narrow them down to about 6 to 10,000. So that's a very high reduction. That's like 3% to 5% of these things. We are saying these are the ones that seem like they should be functional. Let's focus on that and not all these hundreds of thousands of things. So how do we, that's good, but how do we convince that what we have prioritized or what we believe are interesting and functional are really enriched for any function? So one thing that we use to existing data in the literature is these assays called massively parallel reporter assays, where they can take a genetic variant and then put that genetic variant in two different allele forms. Let's say it's like maybe A and C. They put it into a little construct and then see how much gene expression they are driving in an allele-specific manner. So when we looked at these experiments, what we have shown is that our QTLs that are interacting with the promoter, which we call pieQTLs, promoter interacting eQTLs, they are really highly enriched in being validated by these massively parallel reporter assays. This is compared to all eQTLs that are at a similar distance, that are greater than 10 kb. And also, interestingly, compared to something called fine-mapped eQTLs. This is just statistical fine mapping, right? Using the difference in this significant signals, statistically you can prioritize these are better than the other one. So that tells us that the experimental prioritization, or experimental plus our computational method, using that, we are able to prioritize functional variants much better than just the statistical analysis of it. And also, it's not just the experiment, it's the right experiment. If you were to do another experiment called promoter capture Hi-C and prioritize the variants with respect to it, they are still not as nicely validated. This was on one data set. And this is a similar assay, kind of like a variant of that assay. This is, as you can see, that pattern of our predictions being enriched is consistent here. So that I hope convinced you that when you are doing eQTL mapping, maybe it's more important the physical or the actual proximity in the 3D is more important than the one-dimensional proximity, and that gives you much better variants in terms of those that are functionally relevant. So another thing that we have identified, and that's been really nice actually for us to think about it, is that we know that these interactions, as well as enhancers and a lot of things, are cell type specific, right? But in terms of genetic variants, they are the same across different cells, but in some cells they have an effect on the gene expression, in some cells they don't, or maybe effect is strong, but they have an association in some cells and they don't in some other cells. And sometimes in these kind of cases, they may have actually a interestingly weird pattern in one cell type. A G allele on a specific region might correspond to increased expression of a gene, and in the other one it may correspond to decreased expression. These are T cells and B cells in this case. So this is confusing, but when you really map the chromatin interactions and these loops, it kind of makes, and it started making sense, right? So you look at the promoter of this gene. This is the gene that we are showing the expression here. It's an important gene. It's an adapter protein that takes extracellular signal, transfers them into intracellular, signal receivers. So what you can see is that in the T cells, that this promoter seems to be interacting with these enhancer regions. that's downstream, and then that's around this SNP variant. But in the B cells, these loops are not happening, and instead this promoter is looping to the enhancers that's upstream of it. Right. So how can we really functionally validate that what this suggests us is that this is the control region for this gene in these cell types, and then these are the control region for this gene in the B cell. So then again, CRISPR, our best friend, we can use it, and actually you can not only delete or cut or edit the genome with it, but you can tag things onto the CRISPR structure and then target them to a specific region. In this case, we are using that Cas9. It's not going to cut the genome, but it's just going to find a specific region in the genome. But when it finds, it's bringing this big thing called crab complex that goes and inhibits that region. Right? Once we do that, if we target the CRISPR to this enhancer, then we are seeing in the T cell lines, we are seeing an expression decrease, specifically GAP2's decrease in this cell line, but not when you use a B cell line. And then when you target these enhancers, these E3, I think, then you see no effect almost in the T cell expression of this gene, but a highly significant reduction in the B cell expression of the gene. So that essentially on its own tells you that this wiring, this looping already explained now, like why we were seeing an effect well, one way or the other in two different cell types. So this was really exciting. And then one thing that we kind of like get a glimpse of it from this data is that because we have 6 donors, we kind of like whenever we are lucky and then have a division of donors into 2 or 3 different genotypes, we can ask about, okay, I know that this gene is expressed higher in the G allele compared to the heterozygous, and I can look at the promoter histone acetylation, and then seems like these donors would have very similar levels of histone acetylation here. And then the same thing on the enhancer side. But what is different and what is corresponding to nicely to the gene expression is this looping, right? So the donors with the G allele tend to have a stronger loop, and then donors with the heterozygous genome, they tend to have not as strong loops, which really corresponds to these expression patterns. So that started telling us that maybe there are these genetic variants that are really correlating with the looping strength rather than just the strength of the enhancers and so on and so forth. So that's ongoing research in the lab right now. We have new data generated. Hopefully, if we ever meet in the next 2 years, then that will be a complete story, and I can talk to you about it. Another intriguing thing that we have observed is that the non-transcribing but open chromatin promoters. So what does it mean? It's a promoter, and when we say active promoter, generally we mean it's a transcribing promoter, but it's not the same thing, right? The promoter can have the activity markers, but it may not be driving the gene expression. So those kind of promoters, they seem to be serving as enhancers to other genes, right? Which is kind of confusing, that you think a promoter will control that gene, but sometimes the promoter seems like it's helping other genes. It's acting like a switch for other genes. And then actually like this, we are not the first ones to talk about this. A group in Italy and actually Alejandra Medina Rivera from Mexico, they have collaborated and shown that promoters can serve as enhancers, but I think the finding that non-transcribing promoters, the promoters that do not express gene expression, can do that, it was the first in our paper. So here what you can see is 3 different genes, and then this one called HP1 BP3 is a heterochromatin binding protein. So it's super critical. You have to have it expressed in every single gene, otherwise you're not going to establish heterochromatin. But these 2 other genes, they are, at least in these cell types, in these 3 cell types that we are looking at, they are not expressed. There is 0 expression for them, right? But it seems like they do have activity markers and chromatin openness and some loops. Specifically some loops to this expressed gene here, right? So in the T cells, it's kind of like both of them looped here, and then in here, one of them looped here. So what you can then show here is that the if you again do CRISPR, if I target these inhibiting factor to these two promoters, so the nice thing is you are not going to have a side effect. These promoters are already, I mean, these genes are already non-expressed. So you target more silencing things to their promoter, they're still going to be non-expressed. But what does happen is that by targeting these promoters, you are tuning the gene expression on the HPV1BP3 protein. So that's what's interesting, right? So you can use these other promoters and then tune the gene expression of completely unrelated promoter that just loops. So that was just one example, but we have done it for many other examples. This gene where we are able to show that effect is cell type specific. In the T cells you have that loop, and the B cells you don't. And then again, when you target this promoter, that's when you target this, that's sorry, relative expression. Yeah, so this is the one that's expressed. This is the one that you target. So what you see is that in the T cells that you have, when the loop is there, that you have an effect in the gene expression, and in the B cells, the same setting, you do not have an effect on the gene expression when you don't have a loop. So I guess this is just some other examples. I don't want to go into the details of them. But I think then we established another concept that the promoters have these dual roles, and people have known that, right? Enhancers transcribed RNA. So they are not just enhancers, but they also transcribe things. And then promoters, people have been talking about it. They are also promoters, but also regulatory elements. And here we are just specifically showing what are the regulatory elements of, and then validating it using targeted experiments. So the other thing that I want to mention before closing is that, again, like by putting our eggs on the right basket, we are able to like just go beyond that 1 megabase threshold and then identify genetic variants that are like at much longer range. So this is just a visualization of the overall data that if I take out all the variants that are within 1 megabase, these are by and large going to be the most impactful and then highly prevalent ones. But if I did take them out, then we still have good effects across much longer distances, right? Some really nice p values, FTR corrected p values and all of that. So once we did that across our 5 different cell types, what we have found is that we are able to find about 2,000 different SNPs, genetic regions, that seem to associate with expression of genes that are greater than 1 megabase apart. And then this corresponds to about 1,000 genes, 1,000 genes which 70–80% of them are new. So it's about another 700 or so genes that we are finding as eGenes, genes that expression correlates with the variant, in addition to what has been found and reported in the literature. That's maybe it was 8,000 or so eGenes, and then now you have maybe another additional 10% that no one has found before. So that's an important addition, and then that corresponds to about 200 to 300 genes per cell type. So when we looked at them, actually, a good portion of them corresponded to GWAS eQTLs. So these are, by GWAS eQTLs, what I mean is it's a GWAS SNP that was associated to a disease. And now we are finding them as eQTLs for a specific gene. And then in a lot of cases, these were new, right? GWAS SNPs for which target gene is identified by only our new approach. That's about 40 genes here and so on and so forth. So one of those genes that we have done extensive amount of experiments on, it has been this gene called FHIT, FHIT. So that's a cancer-related gene. You need to have expression. That's a tumor suppressor. You need to have expression from both alleles. Even if one allele is silenced, then you are kind of like in trouble, and it's been seen in different cancers. But when you look at FHIT in the DICE database original version, there's no eQTL for it because there's nothing within 1 megabase, there's no variant like within here, as you can see, that associates with its genetic, with its gene expression. So what we were able to find is in about 1.2 megabase away from the promoter, this is the promoter of the gene, we found these regions that have enhancer activity, that have genetic variants, and that are looping in the T cells, not in the other cells, to the gene's promoter. And then when we looked at this specific variance association with the gene expression, as you can see, it's very stringently significant. It's like almost 200 TPM in these individuals and almost nothing in these AA donors, right? So that was very intriguing, finding something that's this far away, and yet they have a very strong effect on the gene expression or association with the gene expression. So what we did, again, like we used different versions of CRISPR, so it comes in different flavors. So one we used is dCas9 CRAB. This is the CRISPR inhibition. When we inhibited this region containing this SNP, again, this is not allele specific. We are just targeting the enhancer. When we do that, we see a nice decrease in the expression of FHIT gene. We can also use these non-cutting dCas9 targeted, but not put this big CRAB domain. Just having the CRISPR sitting on DNA has inhibitory effect on its own. Much smaller inhibitory effect, but yet an inhibitory effect. So we kind of like tiled this region, and then we were able to show that out of these different regions that we targeted, it's this one. It's the one that we have predicted and shown. It's the one that has specific effect in reducing gene expression. So which was nice, but again, these are at the genomic or regulatory element level. These are like targeting, you can think of 100 base pairs or so. They are not base pairs. So then Vivek in the lab has done this extremely challenging experiment that you take the primary cells. Wild type sequence means that donor sequence has a G in it. Actually, like we pick the donors that is GG, so homozygous donors for GG. And I mean, what you do is you kind of like, again, another flavor of CRISPR, but you kind of target the CRISPR to cut from a place. But you give it a template so that once it cuts, it repairs it with the exact base pair you want. So in our case, we gave it A here, the template had A. So it will cut here, and then this G gets repaired or gets replaced by A. So you kind of like the base pair addition here. And then what you see is that we can do it, this efficiency is good, about in half cells we can do that. And then the reduction in gene expression of FHIT through modifying this one single base pair is also really, significant. It's like half again, like per efficiency is 50%, you are seeing 50% decrease. We have done other, it's like extensive again, it's almost tiring and like exhausting me even talking about it, but Vivek has done it. So, and not going to go into details, but we have shown the same things, maybe not so extensively for several other genes where 3-4 megabase away genetic variants, they seem to have really interesting effects on the gene expression of these genes. And then we were able to show that those are likely functional. Okay, so with that, I think I will stop a little bit over time. And then we will try to see if there are any questions. I'm not going to talk about the second part, which is about chromatopsis and cancer, but just want to highlight people who did the work. So all of this work was led by Soria and Vivek. So Soria is my computational postdoc. Vivek is joined. He's done all the experiments. And Vijay and I were the co-senior authors. As you can see, both Vijay's lab and my lab, we do have people from UNAM, and we really love and cherish the contributions that they make to our program. They've been great. And this was the lab before COVID, and we are really lucky to be funded by many different sources and all kinds of research. And those of you who are interested in listening, we are looking at lab members at different levels. I mean, people like your program coming for internships or postdocs or students who come to UCSD for PhD. And all the tools we develop are available through our GitHub. So with that, I will stop and thank you all again for your time and for this invitation, and maybe take some questions. I don't know if turn on YouTube or yeah.
There are many questions. You thought that you were over with the questions from yesterday, but there are many.
All right, cool. Let's do that.
Do that. So Itzel, who was there yesterday too,
yes.
How long does the runtime of Mustache normally take? Does it consume a lot of memory, or what optimization method?
Nice question. Yeah, so it's actually Abbas had a really great implementation. So this hiccups and other methods, they were so extensive that up until recently they were requiring GPUs and GPU clusters and hours of runtime. So right now with Mustache, even the highest resolution data on your desktop, you can run it in a few hours. So it doesn't take much memory, and it's impressive what the kind of implementation.
That sounds really great. Nice. Definitely. Elsa Peters, who was asking, in your experience, which are the most important metrics in order to decide which method is the best for work or works the best for analyzing chromatin interaction data?
Yeah, so it's I think it's a great question. What in our experience, what was clear is that if you stuck on one metric, then you are really not going to get a good evaluation. That's why like we have 3-4 different metrics like reproducibility, recovery of known loops, and then recovery of experimentally validated, and then these other things. So you kind of want to look at all kinds of metrics and then try to find a balance, because it's easy to optimize or maximize one thing, but you might be doing horribly in other evaluation criteria. So you kind of want to get a method that's balanced across different orthogonal criteria. That's what we are doing.
So, but the metric seems like important, definitely.
We have different metrics, but what I'm saying is like performing really high in one metric sometimes comes at the cost of performing poorly in the other one. So that's not a good method that you want to take. You want a method that does either the best or like if you have the best in each metric. That's what I'm saying.
Okay. So Victor Nieto is asking, in analysis like the one related to the identification of functional eQTLs, would it be recommendable to integrate HiChips for different chromatin marks? Since many times the edges are fuzzy,
yeah.
Or are there marks?
It would be, yeah. There would be other marks, but again, these are cost and time issues, right? So we picked one mark that we believe highly represents the active chromatin landscape, but by and large, I mean, you can do like 5 marks, and that would be really nice, but we haven't done that.
So we have a more specific question from Jose, who says, there are conditions in which H3 case 27 acetylation marks are enriched, like Parkinson's disease. Those additional histone modifications are also considered as markers of cis regulatory elements?
Yes, they are considered markers of cis regulatory elements. I mean, there are additional markers that are saying the same. But then again, I mean, yeah, I don't know about the Parkinson disease analogy, but is it all over the K27 acetylation is increased, or in the specific regions it's increased? That's important.
Right. So Diego Arturo, who's asking, which is the main characteristic of FitHiChIP in order to retrieve with statistical confidence mega loops in comparison with other methods?
Mega loops? Okay, so I kind of need to assume, I guess, what they mean by mega loops. If by mega loops,
big ones.
if they are saying distant ones, so then the characteristic is that we have a distance, we have a normalization for the distance scaling. So if you have I don't know 5 contact counts, that's 2 megabase away. That's interesting. If you have 5 contact counts, that's like your neighbor that's 10 kb away. That's not significant, right? So that's the whole idea behind it, that enriches them.
So the next question is from Alex Lopez, who is asking, if we would want to find other type of elements such as trans eQTLs or distant eQTLs. Do we have to consider different variables in these methods, or is it exactly the same?
Yeah, trans is difficult, right? If you want, I mean, if you want to go longer range, then pretty much the same idea will work with deeper sequencing and all of that. But if it's trans, then it's unlikely that there are these trans loops, other than a few very special, contexts or the cell types. We do not have a good understanding of these interchromosomal loops and interchromosomal interactions that seem to be there and consistent enough that will have the trans effect. So if you look at the trans eQTL papers and then the trans effects of genetic variants, they generally break the trans effect into multiple components, like the effects through a transcription factors, the effects through some other mechanisms, and then the looping or physical proximity isn't really the one that's the most prevalent in that sense. So we can do it. I mean, technically, I can just throw in and then I can look at enriched interactions and trans QTLs. We have done that, but nothing clear comes up, and it may not be the most important trans effect. Any? I see others coming. I see.
Yeah, we have another question from Daniela. How did you make the refinement of FitHiChIP spline 1? I mean, which kind of approach did you use? Conservative models to calculate the initial significance?
Yes, we actually in the FitHiChIP paper, we kind of haven't done the refinement part, which squeezes some more statistical power, but we haven't done that. I think they heard about the FitHiC yesterday. That's what they are referring to, but we do not have that in this specific paper. We could have, and they could have given us some more loops.
So then Diego asks, is there any hypothesis for these cell type interactions? I don't know which cell types interactions. Cell-specific TADs, different genome accessibility, cell-specific subchromosomal domains, cell-specific interactions.
Can you say it again? I kind of missed.
Right, it's kind of complicated. I think he wrote.
Okay, then we can take it offline again. I'm happy to answer these things. If they just email me, my email is there.
Yeah, maybe Diego can do that. Yeah. Then it said again, with the potential that these methods have, would you apply them to analyze data of processes such as virus infections?
Yes. So we are applying it to T cell activation. Others are applying it to developmental processes, and viral infection could be one. But then, which cells you are going to look at, right? Are you going to look at the immune cells that are responding to virus, or are you going to look at the cells that are infected by the virus? You can do either way, and there are actually some interesting papers coming talking about how like virus integration, or in general viral DNA being around, tend to change the chromatin organization, this 3D structure.
That's really interesting.
Those are newer papers, but they are really interesting concepts.
Really interesting. So there's another question from Eli Sulvaran, who's asking, when targeting the H3K27 acetylation modification, are we able to map the position of an enhancer? Are there other modifications in different regulatory elements, like silencers or insulators, that can help retrieve different interactions in the chromatin, other than activation or of transcription?
Yes. The quick answer is yes. There are different modifications. We have, if you do it with different methods, you will find different ones. And as I mentioned yesterday, there are methods that capture those silencers and silencing interactions, which is awesome. Again, like here, we are focusing on one side of the story.
And Emmanuel Castañeda, who's asking, evolutionary, what is the importance of the specificity of pieQTLs?
Yeah, it's a good question, but it's not an easy one to study, right? We can look at the conservation of these SNPs, but by and large, these eQTLs are going to be conserved. So whether you could be able to do a QTL analysis, you cannot do it in mice, you cannot do it in, I guess, chimps. So they are conserved regions. These eQTLs are conserved in general, pieQTLs maybe we should compare the conservation of the SNPs that are eQTLs versus pieQTLs. That's a good idea. But other than that, I don't think we can really cleanly answer that question.
And then Hiromi, who's asking, previously I've read that there may be a co-regulation of different eQTLs, trans and cis eQTLs, under the same gene. And then there is something that may contribute to be a specific phenotype. My question is, how do you differentiate each eQTL regulating scope when there is co-regulation?
Yeah, that's a good question. It doesn't have to be trans or you know, cis. It can be two different eQTLs and cis. They may like be regulating one gene, or it may be one eQTL that may be corresponding to the expression of two genes. So we have some of those results in the paper. Once that's online, I think I actually sent that paper to you guys, the preprint. So that has there, but the one concept that came out was that the redundancy, that if a gene is looping to a region, and if it's pretty much the only loop, then whatever variant there is going to have a lot of effect on the gene's expression. If a gene has 100 different loops, that the same gene in another cell type, let's say 100 different loops, including that one loop, then that effect disappears, because there's all kinds of other control switches now that can compensate. So that redundancy is an important concept. But other than that, I'll leave it to the people to look at the paper. My coffee is cold.
So the next question is, what would it mean for disease treatment to focus on the promoter's enhancer with enhancer capacity as a potential target?
Yeah, I think, you know, the whole idea is that enhancers are what are fine-tuning gene expression, and they are doing that in a cell-specific manner, unlike the, you know, the drug discovery right now. I mean, it's evolving, I guess, but generally you have one protein of interest, and then you just try to target it with a monoclonal antibody or something like that, but essentially trying to wipe it out across the board, right? Lots of side effects, lots of non-cell specific effects. Whereas if you find the right enhancer in the right cell type that's tuning the gene expression that you want, then if you have therapeutic target, we don't have it. I mean, there are some talks about enhancer therapy, and there's, I think, a review on that. But once we have those kind of technologies, then that's going to be much, much better, right? Then you will know what you are targeting in when and in what cell type and to what extent. So then that's going to be hopefully like precision medicine, but in the sense that you'll precisely, not the genotype specific.
Very precision.
Yeah.
So the next question is from Carla. Do these cell type specific interaction between cis regulatory elements and promoters have been studied in other organisms like plants or fungi instead of variants of the disease?
They have been, but I do not think they have been studied in the context of specific disease in the plants. So there are studies in Arabidopsis thaliana and model plants. But I am not aware of any that looks at it in the, you know, kind of like a plant disease context.
And then I think it's the last one from this set of people is given from Fernanda, given the good performance of FitHiChIP has shown, are there future plans on using it to identify important eQTLs or pieQTLs in human disease like cancer or neurological disorders?
Yes, again, you need a lot of data for it. But one thing that we may be able to do is we have a cohort of asthma donors or asthma patients, basically childhood and adult asthma patients. So you need to have a highly accessible material, and the immune cells that we take out from the blood is one. And then if you do it for those kinds of cells, you can, like when circulating immune cells are of relevance to a specific disease, then you can do this. It will take a few $100,000, I guess. That's the downside of it. But for cancer and other things, it's a little bit more difficult. The heterogeneity, like let's say you get the tumor cells, they are going to be heterogeneous, and then there's not going to be enough to do a lot of these experiments. That's limited, but you know, you can apply it to, pluripotent ES cells or any other thing that you can think of that you can get from hundreds of individuals, and you can certainly do that. All right.
So I think we are done with the questions, unless there are anything more from Alfredo. Do you know if there are any more questions, or looks like no more? Okay, well, Ferhat, we would like to thank you very much for participating with us again. And it
thank
was
you. Thanks a lot.
And even for non-initiated people like me, I have to say I learned a lot between yesterday and today, so it was wonderful. Thanks
a lot, Yuval. Hope to see you again, and hope to see these students at some point. Thank you all for being so engaged, and then all your questions.
Okay, thank you so much, and we'll keep in touch then.
All right, perfect. Thank you. Have a nice day.
Thank you. Bye-bye.